This program was brought to you by Koller Institute of Venture at Tel Aviv University.
I want to start my lecture in the future. So imagine you wake up in 10 years from now and you feel sick. So you enter your vehicle and you fly to the local metropolitan. There you land at the top of the clinic. A bioengineer would take one drop of your blood, insert it into this small, colorful machine. A few minutes later, 4 letters appear on the screen, A, G, T, and C. This is your genetic information. And a few minutes after that, you go back to the bioengineer, and they design a DNA or an RNA molecule that would fit you, and they advise you to take it 3 times a day after dinner, after your food. Now, if you think this story is imaginative, I would tell you that the most far-fetched item in my story is actually the metropolitan. And you'll understand that by the end of the lecture. So we use a technology called deep sequencing or next-generation sequencing, we and many other labs around the world. And it looks like a big refrigerator, but I will tell you actually how we feel when we use it. So we can enter a human body or a tissue or a cell, and we go fishing. So we cast a very large net, and we catch all the fish in the ocean, and then we bring it back in at the end of the day, and we try to understand it. And in our lab, we look at 2 major effects, or 2 major informations. One of them is genetic, the other one is epigenetic. So genetic is on the left-hand side. That means we could read the entire DNA and content of a human being and look for misprints. 1 letter change in the book of life can lead to a disease or can cause different susceptibilities to diseases or drug medications and obviously many other clinical features. But this is not the only information we collect. We can also collect epigenetic information. And that's on your right-hand side. There you can see that even if we read the book of life, all the DNA, we will not be able to see it because here the message actually from the book is being discarded or it's being crumpled up and thrown away. And this is the RNA message. This is the epigenetics. So we combine genetic and epigenetics in our lab in order to understand health and disease of a human being. And we add other features such as environment and lifestyle to really get deep into the understanding of what happens to a human body in health and in disease. In order to allow all this information to be accessible to physicians and also to each and every one of you, one of the students in the lab wrote a software which we call the Gene Genome, or in short, we call it GeneG. And it can also run on your mobile phone, and it looks like this. GeneG is a redhead in Hebrew, so it's a mischievous red-haired child with DNA glasses, and you can now sequence your DNA and ask it various questions. Of course, you want to know various features, and everyone can ask different questions. This is one question here. You can ask a question about neoplasm, and it wouldn't give you a direct answer, but it would link you to all the current databases in order to interpret your data. Surely this should be accessible to physicians at this stage, but eventually could reach everybody. You can also take your DNA and your smartphone and bump it with your spouse. And here you can try and predict what happens to your offsprings. Now, obviously, this is also done today, just not with smartphones. You would go and you test specific mutations, if you know in your family, and you'll compare it to your spouse to see if there's a recessive mutation that can possibly lead to a mutation in the offspring. So, Tel Aviv University filed a patent on our ideas, and our vision is that we want to provide DNA and RNA information to everybody, and we are at the stage of what questions we want to ask here. We have it as a web server, as a smartphone application, and we have tested it already on about 150 individuals where we sequenced their entire genome, and we wanted to see what information they want to know. This is our patent, which was filed only very recently. There are several competitors, but we believe we have an edge because we combine a lot of information from various types, whether genetic and epigenetic, and we bring it to the physicians at this stage. So we see personalized DNA coming in the next couple of years, and it's not only for personalized DNA information of yourself, it could be DNA of everything. You could be sitting in a restaurant and checking the DNA of your food to make sure you're eating a cow and not a horse. This is 10 years from now. Let's talk maybe 5 years from now and how we use not DNA but RNA, epigenetic information, in order to diagnose brain disorders. So in one of our studies, we went up to olfactory neuroepithelial cells, so biopsies from schizophrenia patients, 20 patients and 20 controls. We took the biopsies. It's a very simple procedure. It's a 5-minute procedure where you can take 1 square millimeter of cells, neurons very close to your nose, very close to your brain from your nose, and you can take it and then you grow it on a plate. So we compared these samples and we asked, are there any RNA differences in these cells? And this is what we found. One of the RNAs was highly elevated in sick patients versus controls. What we found here is that you can take a surrogate cell. So you can take a cell from the body and you can possibly start understanding the disease. We also show that you can have a very simple molecular marker for a brain disorder. In another study on depression, here we went even further. We didn't go to the neurons in the nose, but we went to blood, which is more accessible. And we tested this on 20 depression patients and 20 controls. We measured their effects to drugs. And what we saw, we looked at the RNA level, that one gene really stood out. One RNA was highly elevated and sensitive to the drug, while in the other group, they weren't sensitive. So we were able to divide the responders based on their response in blood cells of these depressed patients. And the final study we're carrying out is on Israeli Defense Force soldiers. Here we're looking at combat soldiers. We have blood samples before and after exposure to stress. We are able to identify an RNA molecular marker in these soldiers prior and after to stress exposure, and we believe we have a marker now that can divide them, those susceptible to PTSD, which is post-traumatic stress syndrome. So our vision is to take surrogate cells, remove cells from the body, and look at epigenetics RNA in order to better understand the disease and possibly have a molecular marker as well. Our patent is not written, is not filed yet, but we're working with Tel Aviv University in order to apply for this, and there aren't any RNA molecular markers yet for brain disorders, and we believe we're very innovative on this end. So in 5 years, we believe that we will be able to use surrogate cells, remove cells from the body, experiment on them, and also extrapolate what happens in health and disease. And 1 year from today, we have something maybe even more tangible now, and this is related to this lady. So this lady is Henrietta Lacks. She died more than 60 years ago from cervical carcinoma. This is with her husband. And this was the first cell line grown in a lab. These are the HeLa cells. The origin of HeLa cells grown in every lab around the world is from this lady. She died from cervical carcinoma. Now we took these cells and we removed the RNA from the cells and we performed deep sequencing on these cells. We ran an algorithm that could take these myriad amount of molecules and build tiny puzzles. And this algorithm, we hoped, would identify her tissue and her RNA, but also maybe other RNA around it. And this is the output of our algorithm. Down below here, you can see that beside human samples, the most prevalent RNA in these cells is derived from human papillomavirus type 18. So this is probably the same HPV-18 that caused her death, caused her disease and her death more than 60 years ago. Obviously, the technology here is extremely sensitive, and our algorithm is able to separate the human cells from all the other pathogens. So what we envision here that you will not only be able to identify your sequences, but also every possible pathogen around you. So our vision is that you can throw onto these deep sequencers any DNA or any RNA sample you want, and then you can better understand your own information, but also everything around you. So also here, Tel Aviv University applied a patent, and we vision that this could be used clinically later on in order to identify the pathogens in these samples. Our edge here is that we use RNA, we use a subtraction, and we have a ranking algorithm that can identify the pathogens around you. So my family arrived to Israel about 130 years ago and lived in tiny villages. Before they knew what happened, it turned into these cities. And maybe before we know what happens, it's going to turn into the metropolitan I spoke at the beginning of my slide. The innovative team in my lab is presented here. They do the work. And thank you for your attention.
This program was brought to you by Koller Institute of Venture at Tel Aviv University.